Welcome back to my course Industrial Biotechnology. In the last two classes, I try to cover the biopolymer and biopesticides, which has a great potentiality in the worldwide market. And coming to lectures, I am going to share two other biochemical products which are already largely in market and has greater potentiality. And one is hepatitis B vaccine production, that is largely, hepatitis is a kind of infection which largely damage our liver, and it is quite detrimental to our health. So, due to the invention of hepatitis B vaccine, now it is possible to protect our health against this particular virus. And another hormones I want to discuss in the coming lectures, that is the insulin, because most of the people, if you look at India and throughout the world, have been suffering from this problem, that sugar enrichment problem in the blood, and this can be rectified with the help of this insulin vaccine. So, first I shall insulin hormone, and first I shall discuss about the hepatitis B vaccine production. Now, before I go to the hepatitis B vaccine production, first let me give you some preamble of that. The hepatitis B vaccine development, and hepatitis B is a kind of virus that infect our livers, as I pointed out, and then the vaccination is the best way to prevent this disease, because that two way we can rectify the disease. One is by using some kind of medicine which can act as which can kill the particular germ, and that is we can use a vaccine with produce some kind of antibodies in the body, and that fight against the particular germ. So, vaccination is the best way of to prevent this particular disease. Then the vaccination is given, if you look at vaccination is given 3 or 4 slots over the 6-month period. In the regular interval of time, this is to be given, and the first hepatitis B vaccine was approved by the United States in the year 1981. So, and if you look at it is World Health Organization, what we call WHO, that lists the essential medicine and most effective and safe medicine needed in a health system. So, that this is permitted by the WHO, that this vaccine is greatly effective as far our health is concerned. Now, what is the hepatitis B vaccine? It is basically, if you look at the recombinant protein subunit vaccine. So, it is a kind of protein that present in the particular virus, and we try to overexpress in some bacteria or yeast bacteria, so that we can reproduce this vaccine, and we inject it so that this antibody fight against this particular disease. The vaccine against the hepatitis B virus is fall under the class of recombinant protein vaccine, and this vaccine are the subunit of vaccine containing only a fraction of pathogenic organism, and it is usually all the pathogens actually they produce this vaccine, because they produce some kind of protein which vaccine that is acts against their survival, and oftentimes that these are the synthetic peptides that represent the protein component that induce the immune system. So, this is all about the hepatitis B vaccine. Now, how this hepatitis virus looks. So, if you look at here, this is how it looks. This is now currently this vaccine for hepatitis B virus is produced by expressing the HBV surface antigen using the yeast expression. So, we use yeast, a kind of overexpression for overexpressing this particular protein, and hepatitis B surface antigen, what you call HBsAg, is one of the viral envelope protein that causes that disease. So, you know that this is how it looks, hepatitis B virus, how it looks. This is DNA, you can see this is the DNA, and this is the lipid bilayer membrane, and this is DNA polymerase. This is where it located, is the last surface protein HB that HBs antigen, this is located here, and medium surface protein antigen, this located here. This is, you can see that the size is, one is small and this bigger, and similarly that small surface protein HB Ag, that antigen also present here, and core antigen also present here. So, in the different portion of the virus it is available. Now, what is the mechanism of action of this hepatitis B virus, that vaccine? The hepatitis B that vaccine is comprises of a sequence of shots mimic the virus that can help in stimulating the immune system of a person. So, that the immunity of the system that improves when we take this kind of vaccine, and as the vaccine enter into the body, it starts producing antibody against the virus, and this antibody fight against this virus. This will not allow this virus to grow, and the antibody produced by the body because of the hepatitis B vaccine gets stored in the body and act in future the person gets exposed to the hepatitis B virus. So, this is how vaccine acts in the body, because you know that if we take this vaccine, then antibody development is there, and that will improve our immunity system and fight against particular disease. Now, industrial production of that hepatitis B vaccine, how it is done using the recombinant DNA technology. We know recombinant DNA technology, we require a plasmid, and in the plasmid we first we find out the gene which is responsible for the production of that particular protein, and then we put it in the plasmids, and this plasmid we overexpress in a particular organism, and then it produces that particular gene, and this gene is responsible for the production of that particular protein. The genes responsible for the production of this HBsAg, this is the antigen, were successfully expressed in Saccharomyces cerevisiae. We know that Saccharomyces cerevisiae is very commonly used yeast, and this has the large use in the industry, particularly for the production of alcohol, and also for baker's industry, but in the genetic technology it is very less used, but particularly hepatitis B vaccine production, it is used mostly for recombinant DNA technology. We use the E. coli for the overexpression of a particular gene. That the yeast system has this complex membrane and ability of secreting glycosylated protein. So, this has made it possible to build a autonomously replicated plasmid containing the HBsAg gene near the yeast alcohol dehydrogenase promoter. So, it has this promoter, the alcohol dehydrogenase, near to this containing particular gene. Now, this HBsAg gene containing the 6 base pair and long sequence preceding the AUG that synthesize the N-terminal methionine, this kind of amino acid that you know that present there. Now, this is how it produce. that this is particular gene that we have, then echo R1, this is kind of with that this is the plasmid that we have, and then that this overexpressed in kind of yeast cell, and from the yeast cells we can culture this yeast cell, we produce this yeast cell, then it reproduce this particular vaccine. So, this is a joint to ADH promoter, that this is the ADH that that alcohol dehydrogenase promoter cloned in yeast vector PMA56. This is the PMA56, this kind of vector that we use. The transform yeast cells are multiplied by tryptophan free media, and transform cells are selected, and the clone yeast cells are cultured for the expression of that HBsAg gene. So, it is similar to the culture of the cells that we have once we overexpress this particular gene inside the yeast cells, then and yeast cells has its own beauty. I know that particularly Saccharomyces cerevisiae has the budding. You can see there is a budding here. You can easily understand that this is Saccharomyces cerevisiae, and they reproduce that on the basis of that budding. Now, if you look at this production process, that as you know, like other fermentation process, that it can grow in a start tank reactor. We know the start tank reactor that is like this, that we have a kind of agitator, and we put the media. We use this contain that start tank reactor to maintain the homogeneity of the reaction mixture. So, we use the soa peptone as a source of nitrogen source, dextrose as a carbon source, amino acid and kind of minerals we use. During the fermentation, rigorous testing is conducted to determine the host cells with the expression vector, because we try to find out that how the desired protein is a protein expression take place, otherwise our purpose will be lost. And this is the industrial fermentation process for the vaccine. This is the obviously that I told you before that the purpose of the major problem with the industrial fermentation process, we shall have to allow our desired organism to grow in the particular reactor, so that there should not be any contamination of the system. Now, if there is a contamination, then the activity of our organism will be lost. So, whole things you have to discard it. So, this is very crucial. The sterility is a very important factor, and when another thing I want to point out here, when you use any kind of vaccine, is directly injected in our body. So, if any infection is there, any kind of contamination is there, it will directly affect your body. So, it should be 100% that contaminant free, because what we technically we call this is a pyrogen free, that kind of the sample that we have, there should not be any, and smallest that living organism we consider as a virus, and it should not contain any kind of other viruses in the sample. So, this is how the organism grow. You can see that this is the organism grow, how the vaccine production take place with this, and this is how the sugar that profile the glucose concentration, how or ethanol concentration, how it decreases with respect to time. And the media composition and the fermentation conditions are aimed to increase the retention of the plasmids. At the end of fermentation, HBsAg is harvested by lysing the yeast cells. So, you have to kill the yeast cells after the growth of the organism, after the fermentation is over, and then it is separated by hydrophobic interaction and size exclusion chromatography technique. This kind of downstream processing, that I want to stress you that that if you look at more or less all fermentation processes look same, because we have now upstream processing where we have media preparation, we have sterilization, medium sterilization, all these things, and then we have downstream processing where we have this purification process. Now, as far upstream processing is concerned, that is more or less all industry is playing the same role. You have to prepare the media, you have to sterilize the media. Only the composition of the media will be changed, because as per your growth of the organism, because different organisms they require different media, and then you have to maintain the sterility. But downstream processing, as I want to point out, that also differ from process to process. As for example, I have already mentioned, nitric acid is separated by the process of crystallization. Penicillin, if you look at it, separated by the liquid chromatographic technique, that extraction technique. And here, that similarly here hepatitis B we use by hydrophobic interaction and the size exclusion, that chromatographic techniques. Now, the resulting HBsAg is assembled into 22 nanometer diameter of lipoprotein molecules. So, it is the very tiny molecules, and this HBsAg is further purified up to greater 99% by a series of physical and chemical methods. The purified protein is treated in a phosphate buffer with formaldehyde and sterile filtered, and then co-precipitated with aluminum hydroxide to form a bulk medicine. This is the downstream processing, kind of downstream processing is adopted just to purify that particular protein. Now, this is how pictorially that can be demonstrated, that this is the virus, hepatitis B virus that we have, and from that we identify that particular antigen producing gene, and this is the plasmid that we have. You can see that you take out the plasmid, and this here you put recombinant DNA is produced, the recombinant this inserted into the yeast cell. This is the yeast cell where inserted. Then in the fermentation process, you grow this culture and produces more from one organism. You produce a several organism, and you produce lot of different proteins out of that. And this is the HPAC vaccine that is that we produce out of that, and that we separated through the downstream processing. Now, if you look that the final product is absorbed in the aluminum hydroxide, which acts as an adjuvant, and lot to lot consistency is thoroughly assessed before marketing the product, because the quality control is very important, because any chemical or biochemical industry, whenever you do any kind of marketing, quality control plays very important role. So, there is the standard methodology that we have through which we maintain the consistency of the product. The vaccine quality is decided by the following factor. First is the potency, then sterility, purity, immunological response, stability, and the protection and longevity. That this all plays very important role as per this that quality control of the process is concerned. Now, that vaccine contains thimerosal, that is a mercury derived preservative at 1:20,000 or 50 milligram per milliliter concentration, as well as aluminum at least then less than 50 milligram per milliliter of vaccine. The vaccine is either sold as a monovalent one or combined with other antigens like diphtheria. We know that we have a vaccine for the diphtheria that can clump together and injected together in the body. Then Recombivax HB1 is a prevention of a hepatitis B infection, was first recombinant protein vaccine for human use and licensed in USA in the year 1986 by Merck. So, this is the double vaccine that has been marketed. Now, this is the downstream processing of the hepatitis B vaccine production. that we told you that how the recombinant yeast production take place, because you have whatever gene you have, this you have to put it in the plasmid, and this you have to overexpress in the your organism, and this is the gene. Then this you put your fermentation process where each fermentation take place. Then you do the harvesting by the microfiltration, addition of protease inhibitor, and cell disruption take place by using the homogenizer. Homogenizer that we make the disruption of the cells. Then detergent extraction by Triton X100, that is concentration about 0.5, 0.2 to 0.5 volume by volume, and clarification by microfiltration, we separate them out, and then again XAD4 for Triton X100 is removal is take place. This is used for the removal of this detergent, and then adsorption to the colloidal silica. We adsorb this in the colloidal silica. Hydrophobic interaction with the chromatographic take place for the separation of this particular vaccine. Treatment with potassium thiocyanate and formulated with alum. This is how this downstream processing of the hepatitis B is taking place. So, industrial if you look at industrial production of hepatitis B vaccine, the common brand, because this is available, it is marketed by the different companies, and the common brand available is the Recom B Vax HB, marketed by the Merck's, and NG-Rix B, this is marketed by GSK, and Elovac B, that is marketed by Human Biological Institute, a division of Indian Immunological Limited, and Genivac B, this is Serum Institute, and another is Savac B, that is another trade name is there of this that hepatitis B vaccine. The India's first genetically engineered vaccine GUNI against the HBV developed by the Santha Biotech Private Limited in the year 1997. And India is the fourth country to develop this highly advanced vaccine, because India is, as you know, India is a vast populated country, and naturally we are also facing a lot of problem with this. So, this can be this is very much required as per our country is concerned. So, we are producing, and we are the fourth the country to develop this high advanced vaccine. So, in conclusion, what I want to tell that hepatitis B is a particular kind of protein that is available in the virus hepatitis, and this this protein is expressed by a particular gene, and this gene has been isolated, and this gene is expressed in kind of plasmid, and the plasmid overexpressed in kind of yeast cell, Saccharomyces cerevisiae, and Saccharomyces cerevisiae when we culture in a fermentation broth, we produce n number of cells, so that we get lot of antigens of this, and this subsequently we purified with the number of downstream processing that involved, and that is marketed in a form that is should be 100% free from any kind of contaminants. Thank you very much.